We've talked about quite many things, and we've sort of transitioned from talking a lot about regression and multiple regression to really talking, well, spending most of our time talking about ANOVA. But I want to make sure we bridge that gap over to really there being the same thing. But as we get into more repeated measures type stuff, the ANOVA approach things becomes a lot more efficient, a lot easier to deal with. So we're going to talk a little bit about the regression approach, and show you sort of how it gets set up and everything. We're not going to spend a lot of time doing it because it is frustratingly complicated and stuff just to do repeated measure stuff, especially when you get into factorial repeated measures and mixed ANOVA. It becomes so crazy. You have to create so many columns to code for everything that it becomes the luster, the shine on regression begins to sort of wear off. So, but I just want to show you a little bit about that. We're not going to spend a lot of time doing it because there's lots of other stuff we can talk about. That's why I said things are about to get weird, because as we're starting to break things up and do stuff, regression stuff starts to become sort of strange. So I said that was sort of a funny thing to put there. But so we're going to talk about repeated measures, and we're going to talk about really combining both together, sort of one-way repeated measures, but also factorial. Now, if you're like me, the first time I heard of there being factorial repeated measures, I thought, well, that doesn't make any sense. How do you have factorial repeated measures? And if you could have somebody repeat through conditions, well, there's no reason why the conditions can't be combinations of IVs that create those conditions that they're repeating through. So you can actually have factorial repeated measures, but they do get a little bit more taxing on the participant, because you're now talking about more and more conditions they have to go through. Okay, so the topics we're going to talk about are the sort of types of repeated measures, some issues and assumptions that come up with repeated measure designs, and then we'll go through some of the analyses. We're going to spend more time on the sort of the one way, but also show you some factorial approach stuff to it. All right. So within subjects and repeated measures are terms sort of used interchangeably. You'll see them used often, even in the same talk. People will switch back and forth and say the same thing. They mean the same thing. It just means that each subject is used multiple times, or in some cases that you have subjects that are paired, like siblings or cousins, or even a married couple and stuff. Sometimes you'll have them used as a pair, and therefore they are repeated, or they're within their linked, they're correlated samples. So most of the time, and then we're going to sort of focus on it here, it's the same subject being utilized multiple times. So subjects cross the levels of the IV. Levels can be ordered like time or distance, right? It can be, time 1, time 2, time 3, time 4. Sometimes, though, they can be multiple measures taken all at the same time. For instance, if you do something like, if you sit down to do an EEG, we have different sites all over, that can be seen as repeated measures in the sense of it's multiple sites being measured simultaneously. If you take 3 different types of depression inventories, you can actually compare across those. You can take the CESD, the Center for Epidemiological Studies depression, and the Beck depression inventory, and you take multiples, and we can actually use those as different repeated measures. But oftentimes, more likely, it is the same subject being used in order across time, like a pre-test, post-test, that kind of thing. Repeated measures designs require less subjects, right? Because we're able to compare conditions or combinations of conditions with the same subjects, and we are able to compare those subjects to themselves across time. So you don't need as many subjects because you're able to reuse the same subjects. That's where they're more efficient, right? Your subjects, participants in experimental designs can often be costly. You might have to pay them to participate. The recruitment process can take time. It can take money. If they get there, there's a time commitment and stuff for getting them to your experiment. So sometimes it's good. It's more efficient that when they're there, to use them for more than one, one condition, one group. So that way they're more efficient. They can be more powerful because participants are being measured more than once. We can identify some of the consistent variability they have and remove it. So more variability can be identified and removed from the error term, and by doing that, we actually make the error term smaller. And you think about these are F tests, right? So anytime with an F where you have, your variability between groups and your variability within groups that we're dividing, anytime you make this part smaller by removing some error from it, right, you're going to make the F bigger. You're going to have more power to find things. Even if your effect is the same, the difference between your conditions is the same, by simply reducing the error, bringing the within groups variability down, we can actually increase F, make it more likely it will be rejected, increase its power. Okay? So in the general linear model, this is remember our one-way between is just we had the mu, which is our population value. We had our alpha, which is our independent variable. And we have epsilon here, or E for the error. So the effect of the grand mean, some IV, and some error. So here, this is where things are different. When we went from between groups, the one-way between groups, to a factorial between groups, what we split up, was that alpha, the effect of the IV got split up into another IV, the beta, and the interaction between the two of them. Well, here we're leaving that alone. It's still one way, so we still just have one IV. We still have the effect of the grand mean, the effect of the IV. But now there's this thing here. This is sigma, lowercase sigma, which is just Greek for S, the subject variable. And then we also have how the IV and the subject variable interact, how subjects may vary from each other differently across conditions. This is like an interaction, actually not like an interaction, it is an interaction between the IV and the subject differences. So we have consistent subject differences, and then these are the inconsistent subject differences. And what we're able to do is take our error term, our regular error within groups error, and split it into the inconsistent part that's sort of changing across the levels of the IV, and then we also have the consistent part, which then is the subject part. So down here, alpha is deviation of scores. All this stuff is the same. Sigma, deviation of scores caused by consistent subject differences, the effect of subject, effect of S. And then we have this interaction of alpha and sigma, the deviation of scores caused by the interaction of A and S, which is often think of it as the interaction of A and S, of the IV and subjects. That's going to be our new error term. So if subjects respond consistently, we're able to remove that out into subjects and use this new error term. This is why this repeated measures version is more powerful, because we're actually able to make this smaller by removing this out. Okay. The assumptions, well, they're mostly the same. We're still assuming that everything's normally distributed, the homogeneity variance, it's from a homogeneous sample. All those things are all the same. The only one that sort of differs is because it's no longer between groups, it's repeated measures, is we can't assume independence of errors. Actually, we know they're not independent. They're the same people, or there are people who are related or matched. We know that they're dependent. We know that they're not independent, so we can't assume this anymore. But in order to track change in mean over time, we still need to assume that everything else is sort of staying the same, that we're actually getting a consistent sort of change across time without other things changing. So what we assume instead is sphericity, like a sphere, but sphericity. And it's called sphericity because if you were to sort of map this out, like it's the idea that the deviations all stay sort of similar, that everybody's staying in a similar sort of sphere in terms of relationships between the variables as you go across time and things. So basically, this is the definition. Variances, so I'm going to read this a couple times. Variances of different scores between levels should be equal for all pairs of levels. Right? Which means you have to have more than two levels. If you only have two levels, if it's just pre and post, then sphericity is not an issue. Really, as you get past two levels, that we start to need to assume sphericity, because the idea is that as we have time 1, time 2, time 3, time 4, right? We're going to, this is, we're going to have subjects taking, going through this subject, whatever, subject N, right? They're all going through this same bunch of time collections across time. This makes it repeated measures. So what we get, and what happens quite a lot, is the farther the time points are, the more people start to drift. They don't stay consistent. Remember, we're trying to identify some consistent error. We want to identify some consistency across people. But if people aren't really consistent, then this approach doesn't really work. So what sphericity assumes is that if I were to subtract, if I get the variance for, say, T2 minus T1, the difference between those two, however much variability there is in the difference, the change, however much people vary in their change from time 1 to time 2, I expect that to be roughly equal to how much the variability that they're going to change between time 3 and time 2, which is also equal to how much they're going to change the variability in their change from time 4 to time 3. But it gets uglier because we're also expecting it to be roughly equal if we start to subtract, say, time 4 and time 2, like all the differences, like it's going to keep going. All those differences are going to keep going, like it's going to be roughly equal to the variability of time 3 minus time 1, right? All those things, you're going to keep any possible pairs of differences. What you want is their variability to be roughly equal. Anyway, questions about that? So you don't really need to know like sphericity backwards and forwards. It's just the idea that differences between time points, whatever the variability is and how people change, that variability should stay consistent for all pairs of levels. So basically, we want variances and covariances between times to stay roughly consistent, right? The reason why it's variances and covariances is because we still are assuming that the variance is going to stay roughly equal, the homogeneity of variance assumption. So adding this in, we want not only the variances, the variability of each of the times to stay consistent, but also the changes, which also means the sort of covariances somewhat. Right, so we want there to be consistent change across time, and we want the variability to stay consistent across time. The trick is, the hard part of this is, and why it's often, like I said, often violated, is the farther the time points get from each other, the oftentimes the less alike they are, the more there's gonna be a lot of variability. So the more time there is between time points, the less people stay consistent. I always think, think about yourself now versus yourself last week, or yourself now versus yourself 5 years ago, right? The further apart you are, the less likely you are to be sort of consistent in your behaviors or actions or responses. So there starts to be a lot of drift apart, and the variability starts to become more and more between time points that are farther apart. And that's why we often have problems if time is a factor, which it often is. We're often going to violate sphericity as an assumption. All right. So take that as much information from that as you can. The idea, though, is that when you do repeat measure design, sphericity is assumed, so you can actually ask for a check for it. All that matters at that point is, in like JAMOVI or something SPSS, is if it's significant, that's bad, just like homogeneity variance. If it's significant, it means that these variances of different scores are actually different, and that could be biasing the results. And if it's not significant, then everything's fine. So we'll talk about that in lab and what the actual test for it is. But they also, knowing that it's likely to be violated, there's all these corrections that you can have added to the output. So basically, it'll judge the amount of violation, like how far you've violated the assumption, and then it'll actually then adjust the degrees of freedom for the test, and therefore the probability accordingly. And if it's still significant after the correction, then people sort of are happy that it still works, regardless of the fact that you violated sphericity. So we'll talk about that in a second. But that's sphericity sort of in a nutshell. it's usually tested with this thing called like Mauchly's or Mauchly's test of sphericity. It's violated. You can use these. It's supposed to be not correct DFs. Use corrected DFs. So in programs like SPSS and in JAMOVI and stuff, you get this Mauchly's or Mauchly's test of sphericity, and if you violate it, meaning that it's significant, there are then corrected degree. There are corrections for the degrees of freedom. Winfeld, Greenhouse Geyser, they're not called lower bound, but that's not in JAMOVI. That they do is they take the amount of violation that is found by this Mauchly's test and adjust the degrees of freedom downward. Right? So you're assuming you have less degrees of freedom, which should make it harder to find significance and basically sort of handicapping. You're getting like a handicap on the analysis. And if you're still able to meet significance with the test, then that's supposed to be good, that you sort of overcome that violation. Let's talk about an example. Let's just dive into some numbers. Talk about here. So I think in this example, I think it's from one of the Tabachnik and Fidel books that is, it's real simple. It's 5 subjects. I think they just had to read, for like 3 months. And the idea is does their liking of the material they're reading get better or worse over this 3 months? I'm not real clear if it's the same book they're reading over and over again or what, but whatever. So it's just some, during 1 month they're taking some measure sort of about their liking of reading, the second month, and then a third month, and they're sort of looking to see that, what's the change over time. The intervention being that just reading, getting people to read. That's it. It's a reading intervention. So here's an interesting thing about this. Looking at this, you should notice right away that this looks very different than when you have a between groups design. The first thing that should stand out to you is that there's this case average. Right? Well, in a between groups design, when these numbers represent different people, because it's 3 different sets of subjects being assigned, it doesn't make any sense to average out those scores because they're from 3 different people, right? So, but because they're from the same person, I now can create this case average, and that's what makes the repeated measure design so powerful, because you can do that. You can actually get a case average and try to figure out, well, all right, if I look at the variation here, this tells me how consistently these cases varied from one another. I can use that to then identify that subject variability and remove it from the error, giving us sort of more power. It's this ability, right, to average them out that makes a big difference. And it's cool because everything's, especially with repeated measures, things tend to be a little bit more balanced because people do drop out. But if people are staying, and dropout does become an issue, that one of the issues with repeated measures is dropout and carryover. Those are 2 big issues, where what happens is you in month 1 can carry over, can affect how you do in month 2. So imagine this is, instead of reading, it was like a GRE prep course. Month 1, you take a practice test. Month 2, you take a practice test. Well, it could have been the prep course, but it also could have been the fact you took the test before could make you do better on the test the second time. So that's sort of carryover. That's specifically a testing effect, which is a kind of carryover. But carryover also occurs in lots of things. Like when you go wine tasting, they usually have you drink things in between or eat crackers or something in between, because the wine that you drink still on your palate. When you drink the next wine, it can affect how you taste it. It can affect how it tastes. It can actually make it taste better or worse. So they want you to sort of clear your palate, cleanse your palate, to prevent sort of carryover from one to the other. So that's a big deal. And then the other one's dropout, because if the same people have to come back each time, well, what's to keep them from just not showing back up again? So oftentimes you do get subjects dropping out. Something that's called subject mortality. That's a bit morbid. Literally, it's like subjects dropping out because they're dying, but they still call it mortality, even though I think I just like dropout sounds better. But anyway, so we got repeated subjects, which means there's subject variability. There's actually this new thing, this subject, the sum of squares for S, we can get by looking, oops, wrong direction, by looking at these averages. We can actually find the variation of these subject averages to identify this sum of squares for subject. And by doing that, we can actually remove it, because the idea that the reason why this works, why this happens, is what do we, when we do it between groups, we assume that sum of squares between groups, sum of squares within groups, I mean, that this is random error, right? So the problem with that is, if we're assuming random error, it needs to be random. Well, looking here, if I have these subjects, these are their averages, it indicates to me that there's a certain level of non-randomness. That there's something about the individuals, like this person consistently scored the lowest or close to the lowest each time. Okay, this person scored consistently highest. So there's something that's not random about how they're differing at each of these time points. Yet when we do this between groups, we're treating all those differences as random. Well, here, this tells us that there's at least a portion of it that isn't random. So when thinking about the consistent versus inconsistent part, this person is consistently the lowest. However, here, actually each time they were tied to lowest, and this time they weren't even the lowest. This person actually was lower, but on average they're the lowest, but they do differ somewhat at each time. That's the inconsistent part. There is part of it that doesn't stay perfectly consistent, and that's how we're able to sort of split up the part that is consistent, the averages, from the part that isn't consistent, and sort of make a new error term that's based on just the inconsistent or sort of random variability within these cells. All right, so that's how we're splitting this up, subjects and treatment, A by subjects. The sum of squares S is calculated and removed from the error term, and it's generally not tested. There's no point in testing it. If you found that sum of squares for S was significant, well, that'll tell you that people are significantly different, which is not super interesting. People are significantly different from each other, and it's not, I mean, that's interesting as a question. So we typically don't, and even in JAMOVI, it gives you the values and it doesn't test it. It doesn't give you enough tests or anything. So we tend to focus on A, obviously, and this A by S, because this is our new error term. This gets computed, but we just sort of ignore it, because all we want to do is remove it out. Model complexity increases. We need to keep track of more and more equations. So this is a little bit of sideline for a second, because I switch over when I usually talk about repeated measures to using this dot notation, which can drive students a little nuts, but it's there for a reason. Before, because our models were fairly simple, we sort of can figure out what was being averaged and what wasn't being averaged and what was being left in. So like when we did, y bar j, the fact that the I, we just dropped the I there, well, it tells us we're averaging over the individuals to create a different mean for each j group, 1, 2, 3, 4. And here we just use grand mean as a substitute to say, all right, this is just the mean of everybody. But with the dot notation, you can get the same information out, because there's two pieces of information. We have I and j, I for individuals, j for groups. So anytime I is replaced by a dot, we know that individuals are being averaged over. If j is being replaced by a dot, then the groups are being averaged over. So we can then replace this by this y bar j becomes y bar dot j, because we're averaging over I and keeping the j's. J's are not being averaged over, so I need one for every j group, but the individuals are being averaged over. Here, because they're both replaced by dots, I'm averaging over individuals, and I'm averaging over. So if I do that, that's basically averaging everything, which is the grand mean. So the dots are just sort of placeholders for anything that's being sort of collapsed or averaged over. That being said, it makes it a little easier when we're talking about where we're finding subject differences and that kind of stuff, and what means we're talking about, especially now we're introducing the subject mean to talk about what things are being averaged and what things are being left, becomes then important, and so the dots help in that sense. Let's see if we can tell what I mean here. Okay, the total variability can be partitioned in between groups. Now in this case, between groups is a sort of still between, I call it between groups because we're going to compute it the same exact way we computed it as a between group in the between groups ANOVA. Like we're treating it exactly the same. It's the mean of every group minus the grand mean. So I don't want to call it something else because it's the same, we're going to compute it the same way. Sometimes people will call it measures, or we can just call it A for the IV or whatever you want, but it's still the same between group variance that we're trying to compute from before. We're going to add to that this sum of squares for subject. This is where you start to see the dot notation additioning, because here, this is I'm averaging over individuals and doing it for and getting a mean for each J. Here I'm averaging over each group and getting a mean for each I. Okay, so go back to here for a second. These means down here, the 2.4s, the 3.8, the 6.4, these are your dot J's, because I have, you know, group 1, group 2, group 3, these are my J groups. So these are, you know, I'm getting a mean y bar dot J. Over here, I'm averaging over groups this way. So now I'm getting, these are the y bar I dots, right? Because I need one for every individual. Subject number 1, subject number 2, I'm averaging over the J's to get those averages. Okay, that's what that's talking about. So this is the mean for every individual averaging over the groups. And I'm going to subtract the grand mean from that. And I'm going to multiply this by k. Now k is just the number of groups. In the same way, when I found this average, I multiplied by the number of subjects that went into the average. When I find this average, I'm going to multiply by the number of subjects in that average. You may be asking, why is this one inside the sum and this one's outside the sum? Because unless we're dealing with dropout or other things, the k should remain consistent. We should have one k, because the number of groups should remain the same for every single person. The number of scores they have, again, unless there's dropout, then that becomes a totally different issue. Here, because we can have different numbers of groups. I'm sorry, the n, actually it doesn't make any sense either. The n here, n for each group should roughly be the same too. This really should be outside as well. Either way, if this was just consistent how we did it before, this one, the k is on the outside because it's not likely to change. It's not likely to change either way unless there's dropout. Okay. Because each of these J's now is a condition that each person goes through. You would imagine the n's will be the same, and so would the k's. But whether it's inside or outside depends on whether or not you want to do it one time at the end or do it each time you subtract the square. Could factor that n out and drop the J out of the n. Inside of the parentheses, I now have our old friend sum of squares within groups. This is every individual in every group. So notice there's no dots now. No dots because nothing's being averaged. I'm doing it for every single person. Minus the mean of each group. So this is my within groups variance. If I take my within groups variance and subtract out that consistent error that I just found here, subtract out the consistent error, what's left inside the parentheses is the inconsistent error. Now this could be 0. You subtract out the consistent error that may explain all of that variance, and then you have none left. This could be 0, but it's likely not going to be. So when I subtract the subject variability out, this consistent part, what's left in the brackets is the inconsistent part. So now we've taken our sum squares within groups and split it into S and error. So because of that, I can take my within groups, subtract S, and what I get there is then whatever's left is that error term, our new error, our new A by S. The interaction between subjects and the conditions is our new error term that we're going to use in this as our error term to find RF. So just quickly sort of talk about like where this stuff all comes from. Total and the sum of squares for A are the same. So I'm not really going to cover those. I'm going to write them out because it's exactly the same. Total, every subject minus the grand mean. So we're talking about, 1 minus the 4.2 squared, 1 minus 4.2 squared, 3 minus 4.2. It's going to go through every single score minus to subtract the grand mean to get you a total variability or total sum of squares. That's the same. Whether you're doing it as between groups or repeated measures, these two you calculate exactly the same. There's no difference. So you're just going to take every score around the grand mean for total, And for this, for the effective A, which is our month thing across here, I'm going to take the mean of each level, and I'm going to subtract the grand mean from it. I'm going to square it and multiply by n. So we're talking about 2.4 minus 4.2 squared times 5. 3.8 minus 4.2 squared times 5. Right? 6.4 minus 4.2 squared times 5. That's what you're going to get. And it's the same. You're going to do this whether you're getting a between groups or repeated measures. Same approach. Okay? So let's talk about sum of squares for subject. Sum of squares for subject is, we're now using these on this side. Around the grand mean multiplied by k, and k is on the outside. So k, in this case, k equals 3, n equals 5. Right? So I can find the sum of squares for subject by taking 3 times brackets. Going to be 3.333 minus 4.2 squared, 4.333 minus 4.2, 4. 5.667 minus 4.2 squared, 3.667 minus 4.2 squared. Okay. You know, square, subtract and square all those, multiply by 3, and that'll give you your sum of squares for subject. I don't remember what that is exactly. Oops, I thought I had it here, but maybe I don't. We'll see in a second what that adds up to, but if you subtract and square each one of these, add them up, multiply by 3, you'll get that sum of squares for subject. Questions about that? Where that's coming from? Next one. This whole thing starts off with the sum of squares, like this part here. This the part in the brackets in the first part here. This is your sum of squares within groups, which you find in the same way you did before, right? Sum of squares within groups is sum of each individual minus the group means. So we're talking about like 1 minus 2.4 squared plus 1 minus 2.4 squared, 3 minus 2.4 squared. All the way through to like the last few, 6 minus 6.4, 7 minus 6.4, 5 minus 6.4. You're going to do all that, find your sum of squares within groups. And once you find that sum of squares within groups, you have to subtract out the sum of squares for S that you already found a second ago. And that's going to be your sum of squares for A by S. Okay. Questions about that? What about degrees of freedom? For A, they're going to be the same. We're going to talk about we have whatever levels of A are minus 1. Easy enough. Degrees of freedom for subjects, or how many subjects do we have? This one gets a little tricky. It's not the number of scores, but how many unique subjects do we have? We only have 1, 2, 3, 4, 5 subjects. So here we're talking about our little n, sort of our little n value. Sometimes people just refer to it as S here to not be confusing. It's not the n in each one, but the number of total actual individual subjects. So that number minus 1. So here we have A was 3 levels. So 3 minus 1 equals 2. Right? We had 5 subjects. So that's 5 minus 1 equals 4. Because this is an interaction, right, we can find the degrees of freedom for it Same way we do for interactions by multiplying them. That's going to be a minus 1 times s. And if you'd like it better to be little n minus 1, it's the same idea. It's still however you want to look at it. Because you're doing it once, not 3 times, until you're doing it here, the interaction. So a minus 1 times your subject minus 1. So that's going to be 2 times 4, 8, 2, 4, 8. Yeah. And then this is the same. So these two, the DFA and DF total, are the same. It's just now the total number of subjects minus 1, which is 15, 1, which equals 2. 2 plus 4 is 8. And 2 plus 4 is 6. I can add 2 plus 4 is 6. 6 plus 8 is 14. Okay. I think I was thinking 2 times 4, as I said 8. 2 plus 4 is 6, plus 8 is then 14. So it all adds up together. That's good. Awesome. Once we find all this stuff, so have we gone through and done all that, we can find, these are all the sum of squares. These are all degrees of freedom we found. We can divide to get mean squares. And notice we have 3 mean squares. You don't actually need to technically find this mean square, right? It's not going to do anything with it. The trick is we're now going to use, instead of using what we used before within groups, we're going to use this mean square, which is the same, but now we're using this error, which is going to give us a larger f. Okay? Because before, if we treated this as between groups, these two would have been together. And so would these two. So we would combine these degrees of freedom and these mean squares and these sum of squares and have a much larger mean square to be dividing by. So here we're dividing, we are able to get out this consistent variance and isolate the inconsistent variance and use that as our error term. That's where we get that f from. Okay? Everything else is the same in terms of all the steps until the end, where instead of dividing by the sum of squares within groups, you're dividing by the sum of squares a by s, so that interaction term. I'm going to go through this quickly, so we're really quickly running out of time. But with the regression analysis, the reason why regression analysis starts to become just insane when doing this stuff for repeated measures is because if you think about it, we need 14 columns for this pretty simple analysis. I need to have 2 columns for A. I'm actually got to code for S. I got to actually have somehow figure out some coding scheme to compare the subjects to each other to cover these 4 degrees of freedom. And then I need to multiply them to get these 8. And that's what it's going to look like. So it's going to look like this crazy nonsense. So you need to actually code for subjects, because now, since there's an interaction between treatment and subjects, well, subjects is now a variable that we have to code for. So the regression setup looks like this, right? We would have our subjects out in our different conditions. We'd have our y's. We code in some way for A. This one's comparing month 1 to month 3. For x1, and this one's comparing month 2 to a combination of month 1 and 3 in the second one. Okay, so we can decide this. This is planned out. This is what we're really interested in. It's A. It's the effect for our IV, right? But then we got to code for S. How am I going to code for S? I just did it. So I did this first subject compared to the other 4, and I repeated that in the next one. First subject minus the 4. It's the same subject. Right, because subject 1 is the same in each group, so I got to repeat that. So subject 1 versus everybody else. Then I got to leave subject 1 out, and I got subject 2 versus everyone else. Subject 2, everyone else. Subject 2, everyone else. Then I got leaving out subjects 1 and 2, 3 versus the others, 3 versus the others, 3 versus the others. Then I'm leaving out the first 3, just comparing 4 to 5, 4 to 5, and 4 to 5. I had to do all that craziness just to be able to code to be able to, when I really don't even care about this thing. But the reason why I got to code for this is so I can multiply A times this to get the interaction. So I now have 8 additional columns of nonsense, right? Because I can't even really interpret any of this stuff, just so that I can actually then get some regression terms and stuff, so I can add these together and figure out what the overall test is for the interaction. It just seems nutty to me. Like, it's one thing to go through in regression and sort of see how the two are the same, but to sort of continuing to go on to do this, it becomes kind of nuts. I mean, and even trying to do it, like because it's so few subjects, JAMOVI had a tough time trying to run this. But I just ran it as a regular one way, and found very similar. This is the repeated measures factor, got the same sort of everything else is the same. But one of the things that's interesting I wanted to show you this is that JAMOVI and like many other programs like SPSS does, I think SAS2 and other programs that are and stuff, when you do a repeated measures ANOVA, it splits up everything. It'll tell you, here's the error term, I mean, here's your repeated measures factor. In this case, it was month. This is A. Okay? Then here's the residual that goes with A. These two go together. These are the within subjects factors. So this is the inconsistent part, A by S. Then you get another column, another table that is labeled between subject effects. These are the within subjects. Here's between subject effects, and you get another residual. What? A residual here, a residual here. They don't do a good job of splitting up. But the only in a repeated measures design, the only source of variability that is between groups is subjects. This one down here is this residual really is the sum of squares for S, where this one's the sum of squares for A by S. And usually they put the error term that goes with the components along with it in the same table, and then they separate out the between subjects effects into a different table. This is another interesting difference between like SPSS and JAMOVI, is that in SPSS, if you click on post hocs, they don't exist. There are no post hocs for repeated measures. Post hoc tests like Tukey and Chaffee and all those were developed for between groups designs. So SPSS is just like, well, you just can't do them in repeated measures. Where JAMOVI has them, you can actually get a Tukey test and other stuff in repeated measures, where you can't in SPSS. That's an interesting difference. And also deciding on effect size becomes kind of interesting as well, because as a one-way design, eta squared seems to make sense, right? However, partial eta squared, add together the A and the A by S, leaving out S, because we already said like S is, like the consistent variation amongst participants, and we're moving it and stuff. So it makes sense to just use A and A by S and leave S out of the denominator. And this actually is more common. So even when you do a one-way repeated measure design, you'll oftentimes see partial eta squareds reported because of this idea that, look, yeah, we can divide it by total, but total includes subjects, which we already said we were taking out because it's consistent. So a lot of people will then use partials to just use A and A by S as the denominator. It becomes a lot more common that way. So for effect size, because of the risk of a true A by S interaction, meaning that subjects, and we're assuming that any interaction between the conditions and subjects is random. In case it isn't, however, and it actually is a real interaction, it suggested you find upper and lower estimates. So lower limit is just regular eta squared. So you find a lower estimate, a lower bound estimate, where you just take your effect over total. And that's sort of like the worst case scenario, the lower bound. And then you compute partial eta squared, where you look at just at A and A by S, that you leave out S essentially. So the difference here is this has S, this doesn't. This gives you a partial eta squared, and you report it as effect size for this study is between the lower and upper estimates, somewhere in between those two. So here's just to quickly sort of give you a rundown. It's like a example of a factorial repeated measures, where, again, the same 5 subjects, instead of going through 3 conditions of month 1, month 2, month 3, reading a book, the experimenters are interested in looking at the effects of 2 different kinds of books. So you got science fiction novels and mystery novels. The idea being, maybe the liking for science fiction gets better over time, or for mystery, because now you no longer a mystery, you don't like it as much anymore. I don't know. But the idea is that you could have a different trajectory over months reading one type of book versus another. And so what you see is that you have subjects going through 6 different conditions. It's just those 6 conditions are made up of a combination of two different IVs, the different types of novel and the different months. Okay? But you can still find over the whole course of things the subject averages, the grand mean. You can get means for each individual cell, but you also have to get means for combinations of things. So look how that works. So what effects will we have? Like quickly, if you're doing this as a researcher, what kinds of things, what kind of effects might you actually identify? The things you're actually trying to interested in. Differences between what? What are the effects? Two main effects of one interaction? Maybe. There are two main effects. What are the two main effects? Say it again, in month? Yeah, so we got novel and month. Those are the two main effects, right? What about interactions? There's actually multiple. So what was the interaction you were thinking of? So we have a novel by month interaction, and that's usually an interaction that we're probably interested in. What other interactions do we have, seeing as how this is repeated measures? I'll give you a hint. We're going to have subjects by novel as an interaction. As our error is one error term, and that error term is going to correspond to that main effect. You get where I'm going? We're going to have, what other one of these we're going to have? Subjects by what? How subjects interact with month. Yep. And that's going to correspond to that effect. And then what's the last one? Subjects by month. The interaction with subjects and the interaction term is also different, also potentially different. So we can also use that. So instead of like with between groups, no matter what the effects were, main effects and interactions, we used one error term. Sum squares within groups, or S with an A or whatever, or sum squares error. Here, every single effect is going to have its own error term because the error term is how subjects interact with that effect. So we're going to have to compute a different error term for every single effect that comes out, which makes this insane. Makes it much crazier, right? That's why I said, things are about to get weird. It's weird this way. So you're having different effects for different things, and it becomes messy, very messy. All right. That being said, I'm going to, by the way, in order to do that, like if you're trying to do it by hand, it's even crazier because I have to remake this data. I have to take and make the data. I need to collapse these three on top of those three so that I can then compare months. Meaning I need to take science fiction month one and mystery month one. I need to collapse them so I can have a single month one. And I need to lay these on top of these in terms of average everything out so I can then compare those. I also need to lay month ones, month twos, month threes together. Sorry, I need to collapse all these months together and those months together so I can compare science fiction and mystery. It becomes crazy. You gotta reorganize the data in lots of different ways to create the means and things you need. So instead of thinking about that, let's think about where all this stuff sort of comes from. So in a factorial between groups, we said everything anchors on the grand mean, plus you have the effect of alpha and beta, which are two IVs. Then you have the interaction between alpha and beta, and you have epsilon. Okay, awesome. Now down here, you got those same effects, alpha, beta. It's just now you also have the effect of subject, how alpha interacts with subjects, how beta interacts with subjects, and how alpha and beta together interact with subjects. That's why every single effect, alpha, beta, has its own error term. The interaction between alpha and subjects, beta and subjects, and then the interaction and subjects. Everything gets its own error term. It becomes kind of crazy. That's how it expands out. I'm not even go into this nuts and get into details because we're running out of time, but you can see you got the effects of A around the grand mean, the effects of B around the grand mean. Here's the effects of A, B together around the grand mean, minus A, minus B. Here's subjects, every individual around the grand mean. Then you have, when I recollapse everything, I have individuals within levels of A collapsing over B minus the A means, and then subtracting out the subjects. Here is collapsing over A to create the B means minus the B means. So it's every individual in B minus the B means. Subtract out the subject so you can get that. Here is every individual minus the subject minus those two things we just saw, the A by S and the B by S, subtracting all that out to get the A by B by S, to get interaction by S term down here. It just becomes sort of insane. Okay, a lot of things going on. Computing each sum of squares requires rearranging the data to get the interaction with S, and when analyzing the effects of A month, you need to average over the effects of B novel and vice versa. The data in its original form is really only useful for calculating AB and the A by B by S interactions. So just doing this by hand becomes just nuts. especially if you're going to do it as a regression term, it becomes silly. Right? It's how overly complicated it is. So I said regression, it is possible, but ridiculous because you would need I think I said something like 30, but it's really, in regression, you would need 29 X's to code for all the effects and error terms, right? You need all the S by effect interactions, all that stuff. It's just, let's not do that and say we did. I mean, let's not even go there because it's so complicated, overly complicated, that's become not very useful. We can look at degrees of freedom. Again, it's nuts. There's 3 levels of A, so 3 minus 1 is 2. 2 levels of B, so it only has 1 degree of freedom. You can multiply those two together to get interaction. S, there's only 5 S's, so 5 minus 1 is 4. And then you start just to multiply everything. A minus 1 times S minus 1, so that's 2 times 4 is 8. B minus 1 times S minus 1 is 1 times 4 is 4. You got the interaction with S, so it's A minus 1 times B minus 1 times S minus 1. It becomes another 8 there. And then total is 29. All this stuff adds up to 29. That's why I'm saying you need 29 columns, because you need a column that corresponds to every single one of these degrees of freedom. It becomes nuts. With multiple IVs and interactions between S and the IVs, it is a bit trickier to find the lower and upper A squared values, because are you talking about including the other A by S's and S's? All this stuff, it becomes a little bit trickier. So do we use eta squares as a lower bound and partials as an upper bound? Well, it turns out JAMOVI provides a third type of eta squared. It's called a generalized eta squared. So you can get an idea of the different lower and upper bounds. I think what it is you have whether or not S is involved. So let's say in this particular case, you wanted the effect size. You wanted eta squared for A. That would be sum of squares for A over sum of squares for A plus sum of squares for S plus sum of squares for everything else, right? Because it's going to be total. So it's plus B plus sum of squares for AB plus sum of squares for B S. I mean, there's just all this stuff in here. A by B by S. All this stuff has to be in there. Yeah. So then I forgot AS. So also plus sum of squares A by S. Right, it's all this stuff in there because this is all total. Well, and then partial becomes sort of simple because it is just sum of squares for A over sum of squares for A plus sum of squares for A by S. It's its only error term. But there's a middle ground. I can put it over everything. I can do partial, But there's also, and I haven't checked, I just saw this last night. I just updated this last night to include it, where there's this generalized that they have in JAMOVI, I believe, is this. It's just this upper one, A plus sum of squares for S plus sum of squares for A by S. It's just the difference of having S in here or not. But this is closer to an eta squared in the one-way sense. This is a partial, and this is a true eta squared that is everything that, you know, kitchen sink on the bottom in the. So here's, just to give you an idea, like I ran this, I mean, this is only 5 subjects, right? So it's a tiny little thing. 5 subjects over 6 different collections, right? So it's a lot of data, but it's really only 5 actual people. So you see the effect for novel is not significant. And you have this like generalized that sits in between partial and eta. Eta being the true lower, this one being in between, and this one being the larger. And that's true of all of them. Lowest, medium, large. Lowest, medium, large. So I have a feeling that's what that generalized is doing. But notice you have novel, residual. So them being together, this is novel by subjects. Month, month by subjects. Novel by month, this is novel by month by subjects. So every single one of them is getting its own error term. So to get this F, I'm dividing this mean square by its own error. That mean square by its own error. It's a very different approach than we do between groups where everything has the same error term. This one now, all the error terms are separate, and we're getting F's based on different error terms because the interactions are different between the subjects and the effects every single time. And then spit off by itself, even though they could do a better job of labeling this, there's another residual down here that's in this between subjects effect, where this is the sum of squares for subject that's off on its own. Right, I also asked for this sphericity test. Notice that for novel, it says NaN, not a number. All right, there's no probability here. Why? So he basically doesn't compute a test for novel. That's because novel only has 2 levels. The 2 levels, there's no need to test for sphericity. It's only once you get to month and month by novel, there's more than 2 levels that it actually does the test. Not significant for month, not significant interaction. So this means that the sphericity assumption is met. Everything's good. All right. And you also see that the greenhouse geyser and Winfeld, these things again are adjustments. You actually use these numbers to multiply by the degrees of freedom that you would use. Like for month, for instance, you would take this 0.831, multiply the month degrees of freedom by this to sort of to shrink it down some, and compute the probability based on that. Actually adjust for the fact that sphericity was violated. But in this case, we don't have to because it wasn't significant. Questions? I think that's it. Yeah, it is.